Hi, I will introduce you to the Mutaframe project. Now, in your body you have DNA, RNA, and proteins. And DNA basically are the blueprint for your body and my body, whereas proteins are the molecular machines that make cells work. Now, the genome information, so the information about your DNA or my DNA, has become extremely cheap in the last couple of years to determine that information, and that has led to an explosion of data. And what that can lead to is personalized medicine. So the difference between you and me could mean that a drug works for me but not for you, for example. But how do you address that? Now, in terms of how it works in your body, if you have a patient who is normal and a patient who has a disease or for whom a drug doesn't work, they will have a mutation in their DNA. And this can mean that the proteins that are written off from the DNA are different. And what we are looking into with this project is to see how this mutation changes the molecular phenotype of the protein and how that might influence a diagnosis or, for example, the way a drug works in the patient. And this is what Mutaframe is all about. And now you will see some demonstrations of the website and how we try to provide this kind of information to medical practitioners so they can better understand what mutations mean at the protein level.
Many genome browsers and variant predictors provide an input field to query mutations. Mutaframe takes this a step further by providing an interactive sequence display that allows the users to navigate through the sequence and pick the mutation they want. Already at this initial step, you have access to useful information such as eminent sequence patterns or amino acid counts. Getting mutation information is done through entering UniProt accession names. You can view the retrieved list of available proteins from the drop-down menu and select the one that you want to display the sequence. Using the interactive display, create the mutation you want to retrieve info and click the Submit button. Once the data is retrieved, you will be provided several layers of information with varying degrees of detail. The coarse chemical properties of the amino acids involved in the mutation. Similarity overview of the amino acids provide breakdown of this comparison into several metrics. The overall Diogen2 prediction score for your mutation and its breakdown into several features. The machine learning contributions that allows you to peek into features that were particularly important for your mutation. Raw scores of input features that enables researchers to compare how the machine learning process behind Diogen2 transforms underlying data. A unique and interactive distribution display that enlists all the mutations that can exist for your protein and their corresponding prediction scores. And a heat map representation of all the mutations that provides essentially the same data as the distribution but from a different perspective. Have full control over your mutations by changing the bin size or simply revealing all data points. Each data point is represented by a single object with value, index, and mutation label. To assess a p-value for your reports, simply click on the curve icon to interactively measure p-value at a given location. Control what you see using the zoom options. Peek through the protein to see how the mutations are distributed throughout the sequence. Export your figure for a publication or simply have access to the underlying data in various formats. The main strength of Mutaframe is not only its tech stack but the vibrant group of researchers and developers behind it working at IB Square. IB Square is an inter-university institute of bioinformatics in Brussels between VUB and ULB, and at IB Square developers make sure the tools meet the standards and are individually designed for its purpose. We also experiment with new ideas to bring the users an enhanced experience in data access and visualization, and constantly iterate over our existing tool base to boost both our publication record and websites. We are planning to migrate our impending tools to Mutaframe project as it gains more public traction. So join us in our effort to redefine genome research. The future of variant analysis is now with Mutaframe.